So hello, everybody. Welcome to our second hackathon teaming meeting for the summer CompuCell workshop. I know I've gotten emails from some of you that you plan to present your ideas today, which is great. But first, I just wanted to go back over some of the information concerning the hackathon for people maybe who weren't here last week. First things first, I'm Hayden Fennell. You've been talking to me here and there, helping to organize the workshop. Presenting here today with James Glazier. He's also been organizing the workshop and is in charge of things here. Just some notes, the slide deck is available at the link here on the slide. You can submit any questions, concerns, or suggestions you have via the chat at any point. There will be plenty of time for discussion at the end of this meeting today. But any questions you have along the way, feel free to put in the chat. One thing to note is that, we do have our Slack channel up and running. If you have not joined the Slack yet, please do that soon. I'll go through and post these links at the end of the presentation today, so that you have access to them through the chat. This presentation is being live streamed and recorded. Just a heads up for everybody so that you know going in that this is being recorded and streamed on YouTube currently. And with that, I think we can get started on talking about hackathon details. There we go. Okay. So again, just to go over the basics of the hackathon will take place after the workshop over the weekend on Saturday, August 6th and Sunday, August 7th. Talk about the schedule a little bit in a couple of slides. Today is our second of two hackathon teaming meetings for you to present your project ideas and potentially spend some time finding people to work with. Today we'll have a chance for people who want to present to take 3 to 5 minutes to present their idea. We do have a number of people who want to present today, so we'll have to be a little bit strict about that 5-minute limit. Once you've finished today, please do upload your project pitch to the shared hackathon folder if you have not yet. And then by the end of this week, we will sort of select a preliminary list of projects that we think are feasible, or we will help scope projects if you have an idea that maybe you think is a little bit too big to do in the 2-day time limit. We can help you with project scoping over the course of this week. For organizing teams, we wanted to kind of help people find groups that they may want to work with this year. At the beginning of the workshop, we'll ask people to choose the top 3 projects they'd like to work on. And then we will put together a list of recommended teams by the end of the day on August 2nd. We're not going to necessarily enforce those teams, but we do want to kind of give people a little bit of guidance if they're not sure exactly what they want to do or who they want to work with yet. If you already have an idea of who you want to work with, or you have a team coming in, that's perfectly fine. We want people to do what they think will be productive. But if you do need some help finding a team, we're here to work with you for that. And you can also work through the Slack channels to find people. So the Slack is going to be a good place for hackathon teaming over the next 2 weeks, because you can have more informal discussions with your workshop mates over the course of the workshop there. Each day during the hackathon, we'll have check-ins in the morning and at noon with the whole group. In the evening, we'll have people give a brief report of how their project is going so we can give you feedback on the next day. And then by the end of the day on Sunday, we'll have you give short oral presentations of your project, how it went, challenges you faced, things like that. We can talk more about that later on. And then we will be available after the hackathon to help people with their CompuCell projects if they are interested in pursuing them after the hackathon. We've had a number of people actually start a project in the hackathon that went on to become used in a publication. So we're always happy to work with people after the fact if you have an idea that you think is worth trying to turn into a publication somewhere. Or if you're just interested in following up for your own curiosity, we're also available to help with that. Some expectations for the hackathon. We do want you to come in with an idea of what you want to model and why, or at least what project you may want to work on. You don't necessarily have to have a project idea to participate in the hackathon. We will help you find a group. Sorry if there's some background noise. My rabbit is digging around in her cage. Hopefully you can't hear that. But we also want you to bring a shareable description of yourself and your skill set or background. Again, it's totally fine if you're a beginner. We just kind of want to get a feel of who knows what and who is available to provide what skills to what project. So some sort of, again, shareable description, a document that we can look through and help find a group for you if you're not sure who you want to work with yet. Expectations you can have of us is that over the duration of the hackathon, we will be available for questions and assistance when it's needed. Again, this hackathon is here to help you get started on your project, and we're happy to help you with anything you may need over the course of those 2 days. If you do intend to participate in the hackathon, we recommend that you take some time to look at the Rosalind Python beginner exercises. We think that you should be able to complete exercises 1 through 5 in order to be sort of optimally successful over those 2 days of the hackathon. So yeah, something to take a look at there to kind of give yourself a gauge for whether or not your Python is maybe where it needs to be to successfully complete a hackathon project. If you don't have an idea yet, but you have a feeling that you might want to lead a hackathon project, we have some project ideas in the form of publications that we think can be replicated using CompuCell3D. This is in the hackathon folder, and again, I'll share these links once I'm finished here. But this is just a list of publications that you could replicate using CompuCell. If you don't have an idea of your own, but you would like to have something to do in CompuCell, these are a good place to start. Again, for hackathon teaming, post your hackathon ideas in the hackathon channel in the Slack workspace. Get people talking about what they might want to work on, what their skill set is. Please also add a brief description of your idea to the shared Google Doc. So in addition to uploading your slides, we have a shared Google Doc where you can fill in your project idea with some details there so people can see that kind of in perpetuity over the course of the workshop. And do upload your project pitch to the hackathon folder if you haven't yet. The lightning talk template, since this is our last hackathon teaming meeting, I guess you don't really need the link to that anymore, but it can be found in the folder as well if you'd still like to put together a project pitch and upload it to the folder after this meeting. As far as the schedule is concerned, so for day 1, we'll spend the first hour finishing up with organizing teams. Again, we want to have most of that done before we come into the hackathon, but just formalizing the teams before we get started for that first hour. Then you'll have your first morning work session. So a 3-hour work block. The idea here is to get set up and get your initial model roughed out. We'll do a quick check-in, followed by afternoon work session and then first day reports. Day 2, we'll start off with just a quick check-in with the whole group. And then you'll have a large work block to work on your model, do another check-in at lunch, work block in the afternoon, and then final reports and discussion. So most of it is just going to be work time where we will be available to help as needed with some kind of check-ins and reports surrounding the work blocks. So that's it as far as hackathon details are concerned. Are there any questions about any of that before we move on to our lightning talks? Yeah, I think this is going to be a review for a number of people in here. But we do have some new faces in the participant list, so feel free to ask any questions if anything was unclear. Okay, so it's,
I guess everybody's eager to get going, which is
Yeah.
good.
All right, so just with if you could use the raise hand emoji, I know I've gotten emails from some people, but just everybody who would like to present today, go ahead and raise their hand so that I have an idea of how many people we're trying to fit in today.
Quick question. If we presented last week, should we present again?
You can if you like. If you don't mind, could we ask you to go maybe towards the end?
Okay.
So the people,
Perfect.
just to make
Thank
sure we
you.
have time for people who haven't presented yet. And if we do run out of time, I'll make sure to link everybody to the previous talk so they can go watch the video. I think we should have time today, though, it looks like. So we have 5 so far. 6, excuse me. All right, 6 presentations. So, and then one more because we've got somebody joining at around 1:15. Okay, so then I don't want to take up too much more of our time so that we have plenty of time to present and discuss. So like I said, we'll need to be a little bit strict about that 5-minute time limit. I will keep tabs on the time for you guys and I'll give you a heads up when there's 1 minute left. It's okay if we go over just a little bit, but again, we'll try to stick to that 5-minute limit. So who would like to go first?
I don't mind going first.
Sorry?
I don't mind going first.
Okay, so we'll do, is it Quirine? How do you pronounce that?
It's Quirine.
Quirine, okay. Sorry.
Let me share.
Yeah, I will stop share and you can get started. Liliana and Manny, did you have questions or were your hands still up from saying you wanted to present?
Oh yeah, just from presenting.
Okay,
Thanks.
that's what I figured.
Do you see my screen?
Yep.
Do you now see it in full screen mode?
Yes, we do now.
Great. So my name is Quirine. I'm a PhD student from the Eindhoven University of Technology. And today I want to pitch this project in which I would think it would be interesting to study the signaling between cancer cells and the extracellular matrix. This project is related to cancer metastasis, which is often the fatal stage of cancer progression. And in this process, malignant cells detach from a primary tumor and undergo various steps before eventually forming a secondary tumor. And in this first step of the cascade, the degradation of the extracellular matrix is taking place. There's invasion of cancer cells, and these cells can migrate through the matrix environment. And the cells in the extracellular matrix communicate and change each other. And for example, this extracellular matrix can be remodeled in various ways. For example, by deposition by the cells, but also by modification and by degradation. And these changes in the extracellular matrix can also change the phenotype of the malignant cell because its behavior is dependent on the environment in which they want to move. And the signaling interactions between the extracellular matrix and the malignant tumor play an important role in this first step of the cascade. So the signaling pathways and interactions between cancer cells and the extracellular matrix change the phenotype of both components. And these changes can affect the emerging dynamic behavior of the cell, but also, for example, the mechanical properties. And one of those signaling pathways that's important in this process is the Rho-REC signaling. And it plays an important role in multiple steps of the cascades. So for example, in the beginning, it can lead to a loss of cell junction, but it can also eventually increase the motilities of cells and degrade the extracellular matrix. And in this project, I think it would be interesting to see whether we can understand the transitions between phenotypes of both the cells and the extracellular matrix, and whether we can link this to the underlying signaling process. So this is a difficult question to answer, but we want to make a first step by using computational modeling. So the biological aspects of cancer metastasis are complex because of the many signaling pathways that can happen simultaneously. So for example, a signal could cause cell A to move faster when in contact with cell B, or the extracellular matrix start to degrade when in contact with cell C, but there are more effects that can play a role. And in biological systems, these effects can become complex, interdependent, nonlinear. So we want to use computational modeling so that we can tune individual parameters and study effects individually. So there's already some research about cellular pods model and signaling, for example, here in source 5. So please have a look if you're interested. And I think the goal of this project would be to find a minimal model in which we can identify a limited number of parameters that affect the remodeling of the extracellular matrix and change in cell phenotype. And I think in this weekend, we could, for example, look at this effect on a single cell level. So in this workshop, we're using the cellular pods framework in CompuCell3D. And this framework is suitable for the implementation of various biological components, including the coupling of internal dynamics. And in this model, we can in principle define cell properties and transitions between phenotypes. And I think in this project, we want to model the cell and the extracellular matrix properties of different states, implement transition rates between phenotypes, all based on information that we have on the biology. And if we could set up these simulations, we can find information about the dynamic evolution of the extracellular matrix and motion of single cells and clusters. So in this project, there are various steps that we most likely need to take. So the first question that we need to think about is how can we translate between cell signaling and cell properties? Then we need to identify some main input parameters and start to study this situation on the single cell level. And eventually, if we look at cancer research, we are also going to be interested in the collective level. So here I've added some research that I've been reading when making this presentation. So please have a look at those if you're interested. And then a little bit about me. I'm a first-year PhD student with a background in biological physics. At the beginning of this year, I started to use CompuCell myself, and I already set up some simulations with a focus on cluster formation in light of active cancer cells. And if you're interested in this project, any skills that could enrich the project is, for example, if you have any knowledge about cancer metastasis, signaling pathways, maybe you have some experience with modeling on the biophysical scale. But if you're interested and have any other skills that I didn't think about, please contact me. I can put my email address in the chat. Thank you.
Okay, that was pretty much exactly 5 minutes. James, did you have any quick thoughts before we move on, or should we move on to the next?
It's a great topic, and I think it's a good one for this kind of modeling. And you've already made some progress, so I think that those are good. I'd love to sit down with you in a small group and go over some of the details of some of the mechanisms you want to include and some of the signaling pathways you mentioned, RAC and RO. I'm sure there are others as well. How do you want to model extracellular matrix degradation, these kinds of things? I was trying to look for a paper by Yi Zhang where she did some models of collective migration in vitro tumor assays.
Oh, that's really nice.
But I don't know why I can't find that paper. I was on the students' PhD committee, so I should be able to find the paper. But I noticed, I mean, are you working with Friedel at all?
Sorry, with Peter Friedel.
Yes, because you started with Peter Friedel's, and of course he inspired a lot of the work on this subject.
Yes, so I'm working in a PhD project, which is part of a larger consortium, which he is part of. So yeah. That
makes sense. Okay. I've always found him an inspirational scientist and a teacher.
Yes.
So that would be great to see this. Let's move forward.
Yes. Nice. Thank you for the opportunity to present. I'm really looking forward.
So I think next we're going to have Issy present. She is on her break from the CompuCell workshop. So she needs to get in while she can.
How's it going? I hope it's going really well.
Yeah, it's good. I haven't used a lot of CompuCell yet. I've been focusing more on the CompuCell, so it's interesting to get a bit of experience in a different one.
Great. You could give us guidance on what we need to improve CompuCell how we need to improve CompuCell. Having done both, you can tell Physicell what they need to do better. You can tell us what we need to do better. That's very valuable. Help.
Okay, I'll share my screen. Cool. Can you see that?
Mhm.
Awesome. So I'm looking at modeling epithelial cell crawling. I'm at the University of Auckland in New Zealand. So it's currently 4 o'clock in the morning here, a bit of a time zone difference. So good morning. So epithelial cells, as you might know, are the barrier for most of the body, like the skin, the cornea, and they stop different pathogens from getting into the cells underneath, and stop a lot of mechanical damage from taking place. Most of these cells are really good at self-renewal. So when they are damaged, they can migrate and proliferate, so divide, and cover wounds themselves. And a lot of them have stem cell populations as well, which help them to do this. So the application of this and taking advantage of the ability for epithelial cells to create their own wound healing process is in tissue engineering. So that is creating a scaffold and then implanting it in the body and letting the body's own cells migrate over there and repopulate the area. So my project is based basically on creating these features or these scaffolds in a computational model and modeling that migration and proliferation of the cells on there. So then we can tune what that scaffold looks like, what properties it has to hopefully optimize the wound healing. So I can move this. Yeah, so there are different mechanisms for epithelial wound healing that have been reported. They have a sort of collective purse string mechanism, which is when all the filaments connecting them kind of just draw in, they all contract, and it just pulls the cells around a small wound. What I've seen more in vitro is this individual crawling of cells, mainly as they will come out, have a little wander around, come back to the tissue, and then the cells will just stream out in sort of rivers. So this is a little video of what some of my cells in vitro look like. When you see the bright flashes, that's a bit of mitosis. You can see this. So as you can see, they form a really elongated spindle-like shape. So all of those are my cells. Yeah, so this is basically what I was saying before, is we want to optimize the wound healing so we can look at different tissue engineered scaffolds without all the complexities of doing it in vitro or in vivo, because there's a lot of different things that are going on when you're working with cells, especially human cells. So if we can simplify that down to a computational model, we can look at different parameters individually and see what has the most effect. So I have been looking at CompuCell because, as you see before, the cells are really elongated as they move around. So that shape change of the cells is quite important. It completely changes the spatial migration pattern. So that's what I thought was really important to capture. I'm in my first year of my PhD. So last year in my honors year, I created a cell automata model, which had one cell per lattice space. And this was one of the things that I found really overestimated the healing time, was not having that shape change. So yeah, expected outcomes, looking at chemotaxis and different substrates, so different stiffness changes the migration rate and the proliferation. If we have different surface patterning, that can cause some alignment of epithelial cells, so they will migrate along different ridges or grooves. So looking at that sort of thing in the model. Yeah, outline is just looking at a small scratch assay with some homeostatic cells and looking how they would migrate out of that homeostatic region and into the free space. And then applying wounding to that edge to see how the cell death and the different signals from that change that scratch assay model. That would initially be a small section and then scaling it up eventually to run across multiple CPUs. Just a couple of papers on some background. Just about epithelial wound healing, how scratch assay experiments work. So that's basically you have a monolayer of cells in a dish and you just take a scalpel or even just like little tweezers and you just scratch down the center and then watch how the cells migrate out. And just a bit of tissue engineering with some endothelial cells. Just to get an idea of the background. So my background is biomedical engineering. So I work a little bit with modeling, mathematical modeling, coding, and I'm also working a little bit in the biology. So I've got a bit of a broad skill set, but it'd be good to have people more knowledgeable on the certain things that this project entails. And what I want to include in the future is from my initial play around with CompuCell, when I added the length constraint parameter, I had a lot of fragmentation of the cells. So they didn't like to elongate into the same shape that I've been seeing in my cell cultures. So I'm really interested in seeing if it's possible to replicate that in CompuCell. Also looking at the different mechanical interactions between those cells as they migrate out. Last week I went to the network modeling workshop. We looked at tellurium. So it'd be interesting to see if there is the possibility of communicating data back and forth between a cell simulation and tellurium to get that communication and any differences with like cell uptake of chemicals and the different states of the cells. And I was also interested in combining CompuCell with vertex-based models to see if there was the possibility there, things like PhysiCell to get that epithelial sheet sliding with the mechanics of that, and also that individual cell movement. Yeah, so that's a little bit about my project. If you have any questions, please let me know.
Thank you, that's great. I mean, everyone should feel free to speak up. Actually, the two presentations so far had a lot of commonality because you're both interested in collective cell migration and ECM remodeling. So I think those are good natural projects for CompuCell. Gilberto, did you want to show one of your elongated cell simulations just as a demo of what you could do in terms of large-scale cell movement?
You mean, I have a question for them. What would be the mechanism that she would use for the cell to move in the first place? Is only chemotaxis?
Yeah, there is definitely chemotaxis in vivo, but at the moment I'm trying to look at more of the mechanotransduction. So yeah, it's more that movement into free space. So when I was looking at CompuCell, I had the motility parameter where it looked at the change in the center of mass, and the cells would move preferably in the direction of free space.
Okay, no, I asked it because we have built a model where there is no chemotaxis at all, and the cell moves around. It's already in the papers in the CompuCell web page. Is something in CompuCell reproduces mesenchymal movement, something like, I don't remember the title, but it's something like that. You don't need to put chemotaxis to make the cell move. Okay, yeah. If you want to cell move in some direction, you can use the chemotaxis, but we have a student back in Brazil, Pedro. He is trying to model this chemotaxis action over the cells, and he is showing that actually chemotaxis doesn't make that the cell move. Actually, the chemotaxis make the cell reorientates its polarization and then moves in that direction. There is no pulling force over the cell to make the cell move in some direction. It's about the reorientation of the polarization of the cell. That could be useful for you if you want to contact me or him. We are happy to help you if you want.
Yeah, that would be awesome.
Is that okay, James? Okay, what simulation you want me to show him?
Just show one of your elongated cells crawling.
Right now I am, let me see if there is here one. It's a very, very simple model that I'm building for the next workshop. And I will share my screen here. Let me see where I share my screen. Is this one here? This is a very simple model. When at the beginning, let me put the cell at start. See, I have actually 3 different cells. I start with some squares. And when you put it to run, but just by adjusting the energies and the constraints of the parameters of the simulation, you can make the cell acquire the right shape. And I don't know, you see, this is the model where the cell, see, the cell is moving. There is no chemotaxis at all. Only in this example, we have a kind of external potential plugin. Do you know that plugin? Yeah. That imprints some force over the cell by adding a external potential. You can make the cell move like that. But we have another ways to move the cell. This is an introductory model to to say that there are many ways to make a cell move in CompuCell. This is the first one with the help of the external potential. So this is for the right window, you can see the trajectory of the nucleus of the cell. It's a very simple model
I thought that it would be interesting to see this just because he's got elongated cells, so he could build very elongated cells with this method.
Oh, yes. The elongation of the cells, you can use some other CompuCell plugin to make it elongate, but I think you have to use the links to prevent the cell detach from each component of the cell. If you stretch it too much, the parts can detach from each other, and then you are messy. Okay.
Yeah. Cool.
I
Thank you so
probably
much
have
for sharing
another.
that.
Let me see if I have another model another movie to show you.
Well, I think, sorry to jump in, but we do have a couple people who want to
Ah,
present
okay,
who need to leave at 1. So maybe what we should probably do is get through the rest of the presentations, and then we can have open discussion.
Okay,
to follow up.
she can contact me if she wants some.
Yeah, we'll probably have time at the end, Gilberto, if you still want to show it.
Okay, thank
But
you.
I think
Sorry.
Izzy has to go at 1 too, so so we needed to follow up separately. But so
Okay.
then,
Yeah, I'll message you. Thank you so much.
Okay.
all right, so we've got Manny, and then we'll do Daniel, and then Liliana.
Okay. Hello, everyone. Share my screen. Just let me know when you can see me. Can y'all hear me? Okay, perfect. Hello, everyone. My name is Manny Lopez. I'm not quite as seasoned as most people in here. I just found this field recently. I got my nanobiotech cert from Alamo Colleges here in Texas, and am trying to move on now to get a master's in bioengineering. Reason being will be this presentation. I call it foam sub. This is my transport vehicle. So my project does seem to have quite a bit a lot of the same attributes as others, but with a different scope. So I grew up in California, live here now. My mom died from a stroke long term. So ever since that happened, I've been curious about the inner workings of what causes strokes, right? Or heart attacks. So the problem is I see it up here, endothelial cells. I'm trying to find out what can actually break down these things called foam cells. The foam cells are basically just free-formed, monocytes traveling through your bloodstream. They get attracted to these guys through signal processing. And then once they're underneath the surface, that's when lipids attach to them through the receptors, and oxygen binds to it as well. Then it becomes dangerous, right? And then it starts engulfing the cell, causing big yellow pulp. See, so this is what I'm trying to get rid of. I know Issy was talking about fixing the walls after the damage has been done. What I'm simply trying to do is to remove the blockages completely, to take the lipids, put it back into the bloodstream so HDL can pick it up and take it to the spleen. So this is just a very high level of the artery itself with the monocytes and the lipids before they become nasty. So I'm trying to figure out what signals do these epithelial cells ignore and send out. This is kind of a two-part project. The foam sub, what does it need to be composed of to travel freely and insert itself beneath that wall, and also more signals tracking the lipids and oxygen. So to do this, and use CC3D, I need to study the interactions of different cell types, specifically bone marrow stem cells into VSMC, vascular smooth muscle cells, and then into the foam cell derivatives. So I want to monitor that whole process through the step process and different energies being applied, and just kind of see if I can model some kind of protein ATP binding agent to dissolve that bond. To do so, I'm going to have my device using those signals, kind of like a homing beacon, go to the receptor, insert itself in there, and then once it's in there, it can play asteroids pretty much, just destroy it. So that would be fun. I mean, if I could really control a little thing that could do that. Anyway, so that's the end goal, very high level, to create an ECM. The ECM is the extracellular matrix. So I will be in a lab in August, maybe mid-August, with Dr. Habibi, and she has some bioprinting equipment. I want to bioprint a cell or a blood vessel, and to recreate all of this, to do the whole lipid formation, aggregation, and deregulation. So if I can do that, then I feel like I would have some success there.
Got about 1 minute left.
Yep. How? Well, CCC3D, need the stat output. These are the types of foam cells or cells I want to endothelial, we talked about. I expect to find the ATP binding agent that can do it, and then translate that into a 3D bioprinted matrix. And skills, just anybody that's interested in these kind of things, electron microscopy, linear regression analysis, bioprinting, and some CAD schematic design and logic board signal processing. But that's it.
Okay, great. Thank you. James, did you have any quick comments?
Well, I mean, I think again, we're everyone so far is in this realm of collective migration and ECM remodeling in slightly different contexts, which is interesting to me. I mean, clearly atherosclerosis and its remediation is a big problem. I mean, both it's a big problem because it's a medically significant problem. It's also a big problem because it involves a lot of parts. And so I think your idea of thinking about what you can bioprint, perhaps, and starting out by thinking about what you could model that would be analogous to things that you could bioprint might be a good place to start. The idea of thinking about pathologically ECM and how you could have cells go in, attack it, target it, and then move out of the tissue. It's also something one might think about. How much detail you can do, I don't know, but I think those they're great questions to be asking.
I know Dr. has her own targeted delivery, so it should work well together. But thank you.
Yep. All right, so then we'll move on to Daniel next, then Liliana.
Yeah, can you hear me?
Yep.
Okay. Yeah, so can you see my screen now?
Yes.
All right. Yeah. My name is Daniel Murray. I'm a graduate student at Michigan State University. And then my proposed project will be modeling the zonal effect of TCDD, activated AHR on the circadian clock genes in the liver lobe. So a bit background. So the circadian clock is a molecular mechanism that synchronizes gene expression, physiology, and behavior with a 24-hour day clock. And then the cellular regulatory network of the circadian clock involves several genes, proteins, and then relies on multiple interaction, transcription, and post-translational loop by communication between the central nervous system and then other peripheral tissues like the liver. So the master circadian clock is in the SCN, which is the suprachiasmatic nucleus. And then this synchronizes with other peripheral clocks in the liver, kidney, heart. And then this helps in several physiological activities like body temperature, hormone release, feeding time. And what we want to do is to study the expression of the circadian clock genes in the liver lobule. And then the liver lobule are the building blocks of the liver. So these are like a hexagonal shaped unit with the liver cells, the hepatocytes, and then we have the central part of the lobule, which is the pericentral, and then the portal part of which are comprised of the bile duct, the portal vein, and then the hepatic artery. And several studies have been done to study the expression of several genes in the liver lobule, and then this lobule can be divided into layers where we have the pericentral layer, that is cells that are closer to the central vein, and then the periportal layer, cells that are closer to the portal vein. So the problem motivation is the AHR, which is the aryl hydrocarbon receptor, is a ligand activator transcription factor that acts as an environmental sensor. And then exposure to environmental ligand as this TCDD can lead to liver toxicity and then downregulate the expression of several genes, including the circadian clock genes. And the disruption of these clock genes are associated with numerous pathologies, including insomnia, heart diseases, bipolar disorder, and cancer. But in spite of numerous mathematical models of the circadian clock, the disruptive effect of TCD-activated AHR has not been studied using theoretical models like differential equation approach. And this figure is from a paper in Dr. Shalitishkaus in 2017, where they look at the interactions between a TCD-activated AHR and then the circadian clock network, where we have the clock BMAL, which is the core transcription factor of the circadian clock, activating these clock BMAL regulated genes. So here we have a healthy clock without AHR. And then even with AHR, we still have a healthy clock, but the moment there's a TCD activated AHR binding to BMAL, then we have an unhealthy clock here, which leads to the numerous pathologies that just mentioned. So the model goals is we've been able to develop a mathematical model for the clock. And so what we did was we took an image of the liver lobule from a published paper, and then we used CompuCell3D to derive the P file of the liver lobule, constructing all the cells in each position, and then we derive a mathematical an equation for that. Oh, I don't know why this is not working. Sorry.
Got about 1 minute left, just so you know.
Yeah. So, and then oh, I don't know, my video is not showing. So this was just an expression of the pair gene in the liver lobule. This video is not showing, but what we want to do is AHR is expressed in a zonal way. So you have a higher expression in the central lobule and then lower expression in the portal lobule. So we want to model the expression of AHR in the lobule, and also when it interacts with the circadian clock genes, is it going to affect the expression of these circadian clock genes in the liver lobule? So we expect to see that we will have a lower expression of the clock genes, even though the clock genes are not zonally expressed in this figure. So their expression levels are the same, but we expect to see some kind of zonal expression with in the clock genes as AHR interacts with these clock genes. Okay, so now I think it's now working now. So this is the expression of the clock genes with respect to time in the video here. All right, so feasibility and project plan. So we already have a model, and then the clocks differential equation for a circadian clock network, but we are still looking for how to add the TCD activated AHR interaction to the circadian clock model. And also from one of our collaborators, Tim Zakariski, we have time series single cell data from in the liver lobe. So we try to also to fit this model after. So the data is in two sections. We have the vehicle data set without treatment and then the treated data set. So we'll fit the data set, the model to the vehicle data set, and then the treated data set to estimate our model. So these are some of the resources. So this paper talks about the space, logic time, the zonal expression of some of these genes in the liver lobe. And then these two papers talks about, these two papers.
Daniel, sorry to interrupt, but we're at 6 minutes. If you wouldn't mind skipping this, people can reference those later.
Yeah, and then I'm open to anyone who is interested. That's the end of it.
Sorry, I don't mean to cut you off. I just want to make sure we can get Liliana in too before she has to go.
Sorry, yeah, that's the end of it.
And we can come back and talk more after the next talk, okay?
Okay.
So great, thank you, Daniel. So Liliana, we'll go ahead and have you present.
Okay, great, thanks. Let's see here. All right, does this look good?
Yes. Yep.
Yeah. Okay, great. Hi, I'm Lily. I just finished my first year of my PhD at University of Denver. I'm actually in the physics department, but my research is kind of on the bio side of things. And I just did a systems biology short course back in June at Irvine. I don't know if you guys have heard of that, but I met Lexi Petrucciani there, who goes to Indiana, and she told me about this course. And basically, me and my group at that course came up with this project that I'm going to present, which is like a multi-scale modeling bio project. So it's kind of the background on how this idea came to light. So my project idea is modeling zebrafish ray branching. So one of my partners from that last workshop, Hannah, has been working on this type of research for her bio PhD for like 3 years. So she's kind of the bio brain behind this. I'm coming from more of a physics background and I'm new to CompuCell. Anyway, so basically zebrafish fins regenerate when you cut them off. And they have this interesting behavior where the rays, so kind of the skeletal structure, branches, which you can see right here as the fin grows. And so we're interested in trying to figure out the signaling pathways and the dynamics that cause this branching. So there's both protein signaling involved and these physical cell interactions, which I was thinking might be more applicable to this CompuCell model. So you have epithelial cells kind of running over as migrating as the fin is regenerating. And the idea, the theory is that they adhere to the pre-osteoblasts that are at the tips of those branching rays and kind of pull the pre-osteoblasts in kind of these two directions in these two branches. So this is kind of a model of that. You can see the epithelial cells running over the pre-osteoblasts, pulling them into different branches. So our theory, our hypothesis, our overarching hypothesis is that there's SHH is upregulated because of some certain signal, which causes this adhesion. And these epithelial cells interact with the pre-osteoblasts to create these branches. This is the motivation for this is basically branching can be found in all sorts of different parts of development like lungs, vasculature. So understanding the dynamics behind that would be helpful in lots of applications. So our idea was to create a multi-scale model, both using signaling pathways and cell interaction. I'm new to CompuCell3D, so I don't know exactly what could be done in the program. I was thinking it's more of the cell interaction level. So that's kind of what I was thinking would be doable in the hackathon period, some sort of cell adhesion model between the cells and pre-osteoblasts, but definitely looking for help and figuring out exactly what we could do there. So that's kind of the general overview of the idea. CompuCell could be helpful because it's multi-scale and you can capture those dynamics of both the cells and the tissues. We probably put in multiple different cell types and have interact and get out the dynamics. So in terms of the project plan, me and my partners, Hannah and Alan, who aren't going to be able to do the workshop, I'm kind of the one doing the CompuCell3D workshop, but the three of us together came up with this project idea back in early June, and are hoping to continue collaborating on it, but also definitely looking for other people who are interested in getting involved in this CompuCell side of it, CompuCell3D side of it, and further parts of the project. So open to anyone. I'm personally new to CompuCell3D, so someone who knows a little bit more in that department could be helpful, but also very open to everybody who's generally interested in this application and project. So yep, that's me. Thanks.
Great, thank you.
Yeah, that's great. I mean, branching morphogenesis is a great problem, and it's addressed in a lot of different ways and different modeling methodologies. So I think it's a good place to look.
Cool, thanks.
Right, so I know we had Scott, and then who else wanted to present today? We had one more.
So can you hear me?
Yes, and then James,
Okay.
that's right. So, all right, yeah, Bharat, if you could go ahead.
I hope it's in the full screen mode now.
Yep, we can see
It is? Oh, okay, cool. Yeah, so hello everyone. I am Bharat and I'm from Indian Institute of Science. I'm a master's student there. I'll be presenting my project, which is titled as Multi-Scale Model for Understanding the Colonization of Ovarian Cancer to the Peritoneum. Let me just briefly go over the progression of ovarian cancer because it's not the more usual way how cancer progresses, which is the hematological fashion, needing blood vessels. Epithelial ovarian cancer, which is the most common amongst ovarian cancer, is originated at ovarian surface epithelial cells, where the transformation occurs and they become more mesenchymal, making them more prone to exfoliation. Upon exfoliation, these cells, either as single cells or multicellular spheroids, can then be transported by peritoneal fluid to the peritoneum, which does not require any blood vessels. And these cells, I'm going to talk about cells. We can go on and think the same for spheroids. These cells attach to the peritoneum wall, where they meet the first barrier, which is the monolayer of mesothelial cells. They adhere to the cells. They causes the retraction of the mesothelial cells, exposing the basement membrane. They upregulate matrix metalloproteases, which chew up the basement membrane and exposes the collagen-rich matrix underneath, which tends to be the proliferative site, and they form the metastatic lesion there. So what the question I'm asking here is, and several epidemiological studies have shown that elder ovarian cancer, actually elder women are more prone to this, prone to ovarian cancer, and they're more likely die from it. So the question I'm asking is, how does the aged histological barrier, the the mesothelia, the monolayer, which I was talking about, how does that promotes the invasion of ovarian cancer? I'm an experimental biologist. I've been working on this for 6-7 months now. Here, you see the red cells, which are RFP labeled mesothelial cells. So basically, I have my cell line of mesothelial cells, and I make them senescent using different methods, radiation or chemical treatment, and I obtain confluent monolayers out of them, and then I add cancer cells on top of them, which is shown in green here. And after co-culturing this for around 3 days, what we observe is that cancer cells are able to clear off maximum of the senescent mesothelial cells compared to the small green patches which you observe in non-senescent on your left. Other studies which I have done, including some time-lapse microscopy, have also shown that the cause of this is that, not the cause, but how it happens is that the cancer cells are able to lift off and cause death within these monolayers, which leads to the decrease in the area of my monolayers, which is shown here. So what do I want to model here? So I was thinking if I can simulate the clearance of these non-senescent and senescent mesothelial monolayers by having cancer cells, and to better understand the mechanism and the dynamics involved as to why these senescent mesothelial monolayers are getting cleared easily compared to the non-senescent one. I was thinking to employ that, and then, well, the prediction here is that cancer cells are able to proliferate better within senescent monolayers, as you can see from the figure, very evident. This can help us answer, well, to some extent, why in elder women we do observe an increase in the incidence of ovarian cancer. And a future goal, which actually is very far-fetched, would be to remove these senescent cells from the picture so that your normal cells can divide and make up for the space, and you can have less progression of cancer there, ovarian cancer there. I am sort of new to CC3D, so I could not really think much about how to employ, but I have tweaked around a bit with the modeling platform. And what I feel is that CC3D is useful here because it allows us to add different features one at a time, like contact energy, growth, death, chemical fields. So what I was thinking was that in the beginning, I can just have like a layer of ECM, and on top of them, have these two the same cell type, but senescent and non-senescent ones, and see if there is some patterning involved by varying the contact energy and sizes. Because we know that senescent cells are larger and flatter. So I was thinking that if a cancer cell is able to, clear off a senescent cell, it kind of produces more space for it to proliferate and stuff like that. So I can imagine some patterning involved. And then switching gears, I was thinking if we can add cancer cells at random on this monolayer, and then add the growth parameter where non-senescent cells can divide and cancer cells can divide. Of course, cancer cells can divide at a better pace than non-senescent ones, and my senescent cells will stay intact because they cannot divide. And then the final model, which I could come up with, was thinking of a 3D model. So other studies have shown that these senescent cells secrete a lot of chemokines, which attract the cancer cells. So in a 3D model, I was thinking of having cancer cells on the top somewhere floating around, and then the signaling or using the chemical field or chemotaxis, these cancer cell gets pulled to the senescent cells in the monolayer, and the same clearance phenomenon happens. And because in the videos which I have seen, I've seen that death is one of the factors.
Sorry to interrupt. We're at 6 minutes.
Okay, sorry.
Would you be able to?
Finish it?
Yeah, to go ahead and tell us what you're looking for.
Yeah, I will just
for
finish
group.
the slide and go
Sure.
on. The rest is not really that important. Because in one of the in few of the videos which I have seen, that cancer cells are able to lift off and cause death. So I was thinking of introducing a cytotoxic effect. I'm not sure how exactly to do that, but yeah, basically somehow adding the death within the monolayer. These are the resources, and well, I do have 2 teammates already, and they have some experience in modeling. But yeah, I'm looking up for more teammates, sure, if you find it interesting. I don't really have, sure.
Were you the one that was asking in the last meeting if it was okay to come in with teammates, or was that a different group?
Yeah. I was asking that because
Okay, yeah.
we have worked together beforehand. We're in the same batch, so there is
Yeah,
a bit
that's
of
fine. I
chemistry
just wanted to
there.
make sure that you were the same person and we didn't have, that I
Sure.
wasn't
Yeah,
thinking of
that's it.
someone else.
I'm done, Hayden. Yeah.
Yep.
I'm open to questions or suggestions.
Great, thank you.
Stop sharing.
Did we have anyone else who wanted to present?
Yes, we have James Doran. Is it Duran or Doran?
It's Dawn.
Doran, okay.
I just make a comment, Barat. There are a lot of feedback mechanisms that could be very interesting here. If your cancer cells are more effective at killing the surrounding cells when they're in a cluster rather than when you just have one.
Yeah.
And if the recovery time is longer when you have more senescent cells, then you could get lots of feedback that will give you a switch from recovery to invasion as you increase the fraction of senescent cells.
Right.
And so I think it's a great project to explore. Okay, so let's go on to the.
Yes, if you want to go ahead, James.
Can everyone see the slides properly? Or is it the presenter view? Not sure.
Yep, that's good.
Okay, perfect. Okay, hi everyone, my name is James. I'm a first year PhD student at the University of Bath in the UK. And today I'm pitching for the project trying to replicate a within-host model of TB tuberculosis. So for a bit of background, tuberculosis is an infectious disease that's caused by the bacterium Mycobacterium tuberculosis. In the vast majority of cases that people get infected with it, about 9 in every 10, the immune response is able to contain the disease by effectively walling it in with an aggregation of immune cells surrounding the bacteria in a structure that is called a granuloma. And you might be able to see this in the diagram on the side of the slide here. For the remaining 1 in every 10 cases, the bacteria is able to overwhelm the immune response and spread typically throughout the lungs if it's pulmonary TB, and this is active TB, which is important because if left untreated will typically lead to death in about 50% of those with active TB within 5 years of becoming infected. These days we have antibiotics available, and normally multiple ones are administered over a span of usually at least half a year. But even if successfully treated, the disease can relapse later in life. And even if a granuloma can be formed and latent infection is the end result initially, especially in immunocompromised individuals, the reactivation can occur later in life. So my project will be looking to try and replicate and therefore hopefully try and validate the results of a paper from a few years ago, in which the authors hypothesized that the initial position of the inoculum within a sheet of epithelial cells within the lungs, its initial position would be important in determining the ability of the immune system to contain the outbreak. Specifically, the further away the initial inoculum was from the nearest blood vessel, the less likely containment would be to be achieved, as the immune cells needed to form a granuloma were recruited from these blood vessels and therefore would have further to travel to try and get to the bacteria before they could replicate out of control. The other hypothesis was that dormant bacteria, so metabolically less active or slow growing bacteria, are the ones more likely to be left at the end if the disease is contained or successfully treated, due to their ability to somewhat resist the cytotoxicity of the immune cells that combat them. And they do tend to be the more likely phenotype to be antibiotic resistant. In general, a future motivation for this is my PhD is looking to develop multi-scale models of infectious disease And so the hope would be to try and nest a within-host model of TB into an epidemiological model as part of that project. And if the within-host model was successfully created here, then perhaps look to adapt it to other diseases like COVID-19. Admittedly, different disease dynamics, but it would be good to have an initial base that could be adapted. So the aim of this project would be to try and replicate this model, compare the outputs and see if we're able to provide some validation to the original paper, and try and understand what does cause the different outcomes in terms of latent TB versus active TB and whether granuloma can form or not. Is dormancy important? Are the spatial aspects of the domain important? And then if there's time, possibly try and expand upon the model and add a few more immune cell populations, try a few different initial conditions. So I think that CompuCell would be ideal for this, as the ability of a granuloma to form is clearly a very spatially dependent system, which is well captured here. So we'd be looking at inputting the bacteria, the different immune populations, different chemical fields, and see as a result, do we get latent TB or active TB, or initially latent TB followed by active TB where reactivation has occurred later on. So the original paper that this is based upon, I put the link here. Ruth Bowness is my PhD supervisor, and she's also provided me with the existing code for this model, which I can share to anyone who may be interested. So that's the project really. So a bit about me. My background is primarily in mathematics. I did my master's quite a few years ago now, and I've only just come back to start my PhD. So I have some prior experience in Python. I wouldn't call myself an expert, and my biology education formally ended when I was 16. So anyone with a good immunology background, good biology background in general, expertise in programming is much appreciated. But in general, anyone who has a strong interest in this, I'd appreciate any help, as the hope would be to maybe perhaps publish a paper based on this later on that could contribute towards my thesis. So I thank you all for your time and your attention.
Great. I look forward to looking at that paper of your advisors. I mean, there are these nice published papers of Denise Kirshner's of granuloma formation. And so those should be relatively straightforward to reproduce in CompuCell.
Yeah, definitely. Yeah,
whether they have the right things that you need is another question. But if they do, those are a good place to start, because it's all, as you point out, it's always easier to take a model that's been developed in one place and repurpose it than to start completely from scratch. But I've wanted to see this some TB models using CompuCell for a long time, and of course we've done the models of viral infection in tissue patches in the lung. But those didn't include the idea of granuloma formation. And so I think trying to understand that would be very interesting and practical. How far you could get in 2 days is another question, but it's definitely a practical project.
Great, thank you.
Great. Thank you, James. I think, so is that everybody who wanted to present today? So I know Max, who said you had yours, but I do want to maybe come back. Daniel, are you there still?
Yeah, I'm here.
Okay, great. We did, sorry that I had to cut you off earlier again due to the time constraints, but I would like to come back around for questions and feedback for you if you wanted to maybe throw your slides up again.
Oh, okay. So put it up. I'm coming up. So let me stop sharing and then look for it and then.
Sure.
Okay. Yeah. All right. Yep. And then let me share. Sorry for the delay.
Oh, it's okay.
Yeah.
Caught you off guard, no problem.
Yeah, and you see my screen now?
Mhm.
Yep.
Yeah, so I just wanted
Yep.
to give a minute for James to give some feedback and anybody to ask questions if they had any.
Well, I think they'd be, so have you put the circadian clock model into the cell, into a liver, zonated liver model yet?
Yeah, so that is what I'm showing here. And then from this paper from Nature Metabolism, the expression of the circadian clock genes are non-zonated. So when you model it across the cells, the expression are gonna be the same, like in take the average of it.
Right.
So when you look at the expression from the cells, the central lobule and then the portal lobule, and you average them, it's gonna be the same. But when you model the expression of other genes like AHR, CYP2F2, those genes are zonated, so they are gonna have different, zonal expressions. Yeah. So that is the next stage of the project, to incorporate the expression of AHR into the model and see if, because of the zonal expression of AHR, we also have a zonal expression of the circadian clock gene.
Okay.
Yeah.
Well, that seems achievable, given how far you've already gotten. I think that should be sounds practical to do. And again, I'm sorry, everybody, I'm getting over COVID. And so, well, over is maybe an exaggeration. I'm recovering from COVID. And so I'm still not 100%. I hope by next week I'll be back to normal. Today I'm still a bit under the weather. But that's great. I think that's good. And it'll be nice to see that project develop. I hope we can pair you up with some people to get some additional help with that, because I think it's a good opportunity.
Yeah, we really appreciate that.
Okay, are there other people who want to present, or questions, comments?
So Max, did you want to present again, or would you like me just to forward people to the other video to view, or did you have something to add from last week?
I can just give a brief overview. I'm actually out of my house, so I can give like a 2-minute thing just to save everyone's time, if that works with you.
Sure.
Can you guys see my screen?
Yes.
Yep.
Okay. So hi, everyone. I'm Max Baga. I'm an undergraduate at Emory University, and my background is in infectious disease modeling. I've done a few models with influenza, which I'm presenting on here, and then I've done a bunch of thyroid hormone modeling, and I really want to bring both those fields into my model. So the way influenza evolves is that 2 distinct strains will infect the same cell. And since each strain is made up of these 8 segments, they'll each exchange segments and they'll produce these new viruses. As you can see by this figure, 2 viruses on the right infect the cell. They recombine and form these distinct viruses. And this is what really drives pandemic strain evolution. So what will happen is that in an avian species like a mallard, 2 distinct strains will infect. It'll produce this part avian virus, part mammalian virus, and that can eventually infect humans. And what I really want to model is I want to model this reassortment at that cellular level, see how infections can spread, how different lobes of the lung may support reassortment to different extents. There was a few papers done using CompuCell3D, modeling infection, modeling immune response, and I would like to draw on a few features. in terms of like the actual parameters for the model, I have a few of my own papers where we measured the rates of reassortment, sort of characterized it, and I hope to bring those into the model. And I have never used CompuCell3D. I do have extensive Python experience and modeling experience. So any teammate with any background, I'm open to working with. That's sort of my 2-minute pitch right there. I did a more detailed version in the last meeting. I think that recording's on YouTube. So if you guys want to join my team, you can email me at max.baga@emory.edu and feel free to reach out with any questions.
Okay, great. I think I was thinking about different ways one could model reassortment. And I suppose you could have your virus could have not eight indices to it. And so if you have a super infection, then you could get recombination, reassortment. And then if you need to have some hypothesis about what the infectivity of the mutated virus is, though.
Okay, I'll come up with it. I have done extensive reading on reassortment in humans, and I definitely can draw on that knowledge.
But including just in the kind of tissue model that we did, the very simple one, including the idea that instead of having a single virion, you have 8 different components of the virion, and so each viral assembly is composed of 8 components. I think that might be manageable to do.
Okay.
Again, I mean, how far you could go in 2 days is always difficult, but I think conceptually the idea is reasonable. You could start with just 2 components. You have two multiple strains of virus which have an A and a B component that are different. And you ask, if you have a super infection with 2 components, what happens? And then if you could get that to work, you could extend to 8 relatively easily.
Yeah, definitely. The 2 segment is definitely a great way to start, because HA and NA are usually the 2 determinant ones in humans on how they enter the cell and stuff. So I definitely can start with a simple model with just those 2 segments.
Yeah, I mean, I guess they call it, right, I mean, we characterize it IAV from H and N for a reason, I guess. Not that there aren't plenty of other mutations that aren't important. Okay, great. Well, I think they're all great projects, and I hope we're able to get some good teams together for them. Other comments before we break for today? We've gone about an hour and a half, but we blocked out 2 hours to keep talking if we need to. We also
go ahead and put links in the chat for you before we leave, so everybody has those. And these will also go out in a summary email later this evening with the link to this video as well. But yeah, now if there's any questions or if anybody wants to have any discussion about anything, the floor is open.
Well, not a question. Something which you pointed out about a switch happening between the 2 monolayers, wherein you have senescent and non-senescent. Well, it's just intriguing when instead of having single cells, when I have done the experiments with spheroids, when you have bulk of cells, cancer cells, we do see much more of that sort, even in case of non-senescent monolayers. So I do see the point being, having a switch wherein can it come back or can it recover or not, based on the initial number of cancer cells you have.
Right. Well, I mean, one of the first talk we heard, I think, or the second one, was somebody working with Peter Friedel, who's done all this wonderful work on the fact that much cancer metastasis is collective invasion rather than single cell invasion. And so you could build a very simple model where you have a cancer cell and it has some probability of attaching to the substrate. That probability, if there's a gap in the substrate, it has a higher probability of attaching.
Right.
If it
Right.
hits another cell, it has some probability of killing that cell and attaching. And if it stays, once it's attached, it has some probability of staying attached. If there's a
Right.
hole, there's some probability of it being, of the hole being filled by proliferation of the neighboring cells. And so you could build a very simple model of that kind where if the tissue is attacked enough, if the tissue is attacked just a little bit, it's able to restore itself and get rid of the cancer cells. If the rate of attack on the tissue is too high, then the cancer cells begin to form footholds and spread on the tissue substrate. And so I think
Yeah, that makes
that kind of model can be done. A fairly abstract model you can build pretty simply. Then making it meaningful biologically and parameterizing it is more difficult. But I think getting the basic feedback loops that you talked about and the thresholds that you would talk about, those should be possible.